We all have noticed, or for centuries, we noticed that characteristics are inherited from parents to children. And we can figure out based on the features who is whose children, at least to some extent. And so, what is known is that there is inheritance of characteristics. We don't know the molecular nature. Where is this information stored within the cell or in the body? And in those days, there are several theories that people have put forward, in which include many, some of them just cooked up or trying to make sense, because we should give them the benefit of doubt that because they do not have the information that we have, the science and technology at this point. So it was thought that the sperm contained minuscule human in there, and then when it's impregnated, that grows into a new organism or infant. Similarly, some people had much more complex ideas about which pregnancy will result in a girl and which results in a boy, based on heat and cold and different things and the different environmental aspects that would have influenced the pregnancy or this whole process. Some others, including that of Darwin, had a hypothesis something like this, where he thought that, or at least in his visions, different features or features from eyes or nose and hands and different things, they come into the germplasm. And from germplasm, say we have an egg and a sperm, and then they fuse together, fertilization would occur, and then will give rise to an embryo carrying these characteristics of, for example, the eye color or how the nose should look like, and so on. So if this is the situation, and how did people figure out what is genetic information and how the inheritance happened? And that forms the molecular basis of the whole of molecular genetics. I am Dr. BCMR, Assistant Professor at Shastra. So before going into the different experiments, or some of the experiments, important experiments, we need to try to delineate the experimental setup, because the information from these experiments is no longer new to us. But the scientific thinking and the way people have designed their experiments is what is of importance. So we need to prepare what kind of preparation did they make, what kind of model organism did they use, why did they choose that model organism, or why is that model organism good for this particular experiment, and what kind of techniques did they use, and then how did they interpret it and communicate it. These are some of the things that we actually need to take from this, because the information from these experiments is already we already know it. So the first one is Hammerling's experiment. It's about Estebularia. It's a small, tiny plant-like organism which has the stalk at the bottom of it, and then the root-like structures, and then it has a long stalk and a crown on the top of it. So it can grow to 1 or 2 centimeters also, some of the species. And the important thing is it's a single-celled organism. It has several parts. Apart from this, internally people have known that in the base there is a nucleus. So now there is an experimental hypothesis built. So which part of this organism has the genetic information? Is it the crown? Is it the stalk? Or is it the base roots? So Hammerling's experiment is interesting. It's simple and elegant for that time. He has chosen 2 different species. One is Estebularia granulata, and the other one is Estebularia mediterranea. So they are almost same, except they have some physical characteristics that are different. One of it is green, granulata is green, while mediterranea is brown. And also if you look at the structure of the crowns, one of it is much more folded, the crenulator one, whereas the Mediterranea is something like a blossomed flower. So what the experiment Hammerling has done is he cut it into 3 parts, as you can see here. And he then exchanged the pieces, stalks, onto one another. So the brown Mediterranea got the stalk from crenulata, and the crenulata base has got the stalk from the Mediterranea. Now the question is, what kind of crown will it build? Will it build a crown that is in green in color, or will build a crown, or will a brownish crown will emerge on this part? And in the same way, what will happen on this one? What they found is the stalk, the crown that was built is same in color to that of the base that is shown here. Similarly, on the Mediterranean base as well, you can see what happened was that a brownish crown has come, which is similar to that of, or which is typical of the Mediterranean base. And remember that apart from the physical features, there is one important feature that is different among these 3 parts, is that the base has the nucleus, and that the nucleus is determining what kind of feature, what kind of crown and stalk would form. Similarly, there are also other experiments. This is an important experiment, which is the basis for many modern day technologies such as animal cloning, regenerative medicine, and embryonic stem cell technology, etc. So this is conceived by Sepman and then taken up by Briggs and King. What they did was they have taken an egg, and the egg was enucleated by using UV light. So UV light will damage the nucleus, and then it is no longer functional, or the genetic information is all cleaned up. And then they have taken somatic cells, the epithelial cells, and from there they have taken out a nucleus and then put it into this enucleated egg. And majority of these manipulated embryos have died because of these procedures, which are usually harmful. And some of them have grown to a certain stage, and then they were abnormal, and then they perished. And very rarely, or some of them, have grown on to give rise to a complete organism, like in this case. What is interesting in these experiments, or what they have done, is the characteristics of this frog, new frog that has come, had the characteristics of the nucleus from which was implanted into this enucleated egg. It was not of the characteristics that were coming from the egg, which would say again that the nucleus has the genetic information. And the nucleus is a complex cell organelle itself. It has multiple things. It has proteins, nucleic acids, lipids, and carbohydrates as well, maybe to different ratios. Out of which we now have to figure out, chemically, what is the chemical nature of genetic information? Is it proteins? Is it nucleic acids, lipids, or carbohydrates? There are, out of these 4 different biomolecules, at least 2 of them are very big contenders, because they are polymers, and they have they have diversity in their components. So lipids are not much of a polymers, but carbohydrates such as starch and glycogen are polymers, but they are made up of the same molecule. If it is same molecule, they cannot mean different things, right? So if some molecule has to qualify to be an information, then it is important that there is diversity in their components, same as how we have in English language, for example. We have 26 alphabets. Different combinations of these alphabets will give rise to different words. Different words put together will make meaningful sentence, and then paragraphs, and then whole essays or songs. So if a molecule has to qualify to be as an information, then it must have diversity in their components, subcomponents, and then it must be a polymer. So then nucleic acids have 4 different bases, and so you can possibly make a good number of words with them. Whereas proteins have 20 different amino acids. That means the diversity is more, almost similar to that of English language. So they should be much more well qualified to act as genetic information. One thing we have to remember, however, is that any information or the genetic information that we discuss here should be inheritable. That should be able to make a copy of itself. I hope I leave you with something to think about, and why nucleic acids are the genetic information and why proteins are not. Thank you very much for your attention.